into the cornea, especially if the affecting agent, Pseudomonas for example, is resistant. Fortunately in the UK, for example, the amount of resistance is generally relatively low, but in places like India and elsewhere it's becoming a real problem, with at least 25% and sometimes climbing up to about 50% of isolates being highly resistant to the different drugs that we use. Pseudomonas comes in two types, or it comes in many types, but two main types. These are called XOU or XOS. XOS, you can see on this side, these are called invasive strains. If they contain this particular gene, this allows them to invade cells, for example, corneal epithelial cells. If they, instead of containing the XOS gene, they contain the XOY gene, and these tend to be mutually exclusive, they then become a cytotoxic strain and kill cells like corneal epithelial cells and our white blood cells. So we've been doing whole genome sequencing of many strains of Pseudomonas that we've isolated from keratitis in Australia and India. We're looking for the presence of XOU and other antibiotic genes that are associated with antibiotic resistance. We look for mutations in these genes, and we look to see how they relate to antibiotic resistance phenotypes, and then the frequency of the resistance between the XOU and the XOS, and the frequency of possession of genes or single nucleotide polymorphisms in chromosomally derived genes that are also involved in resistance. So you're going to see a series of slides here. On the left-hand slide, you can see the strains with XOU and the strains with XOS. If they're in black, it means they're resistant. If they're in blue, it means they're not resistant, they're sensitive. And you can see this is an ongoing process, so we haven't yet filled out all of these, and MD just simply means we haven't got around to testing these yet. But you can see for fluoroquinolones, hopefully, that the XOU strains have much more black than the XOS strains, and so much more commonly are resistant to fluoroquinolones, like ciprofloxacin, levofloxacin, or moxifloxacin. And they tend to be resistant because they have mutations in the genes called gyrase A and PAR-C, which are involved in making DNA properly, but also in other genes, for example, in a gene that's associated with fluoroquinolone resistance and other resistance, QNRVC. And that just gives some data around this. You can see there's significant differences between the XOU and the XOS genes in resistance and in the genes that they possess, as I said. For aminoglycosides, again, hopefully you can see, again, the XOU has much more black than the XOS. And so, again, the XOU is much more commonly resistant to aminoglycosides, like gentamicin. In particular, you can see the data for here. And that's associated with the possession of various genes on plasmids or even chromosomally integrated that destroy the aminoglycosides as they come into the cell. And a similar, or not quite as apparent, but a similar problem with the beta-lactams as well. Again, XOU is, hopefully you can see, either black or grey, meaning it's intermediate resistance or sensitivity. Much more black and grey in the XOU than in the XOS, especially for imipenem in this case, and for some of the genes that again come in on plasmids or other mobile genetic elements, or indeed single nucleotide polymorphisms. Again, you can see the data here of the differences and the significant differences between the possession of genes or the single nucleotide polymorphisms. So it does look like the possession of XOU is more common in ocular isolates of Pseudomonas aeruginosa, and if they possess that, they're much more likely to be resistant to fluoroquinolones, aminoglycosides, and beta-lactams. And of course, that's a problem because we tend to use those to try and treat these diseases. Association and causation, I think we need to do a bit more further research to understand whether the actual genes that we see are the ones that are really associated with the resistance. Thank you very much for your attention, and we can have maybe one question if there's any burning questions. Who wants to give me a round of applause?
Did you check the efflux pump?
Did we look for efflux pumps? Yes, we looked for efflux pump genes as well, and some of them definitely do. And again, XAU tends to have more efflux pump genes than XOS as well, so yeah. Good, we'll move straight on. It's in front of me. Nick, you're up next, and Nick's going to talk about identification and quantification of Acanthamoeba species within seawaters at coastal lagoons in Australia.
Thank you, Mark. I've got a map of Australia up here, and the part of Australia that I'm talking about, just over on the left, is this tiny little slither here, which is about 80 kilometres north of Sydney, and it's in a subtropical area and a very popular holiday spot in New South Wales. And from this study that we published in 2018, we showed that if no one wore contact lenses during water activities such as swimming, we could reduce the number of Acanthamoeba keratitis cases by 1/3. And that is because Acanthamoeba, here shown as a trophocyte attaching to a contact lens surface, likes to bind to contact lenses, and that's both in this trophocyte form, the active form, and the inactive cyst form. And just a reminder is this is what it can end up like on the cornea. So in this study, we were looking at the incidence of Acanthamoeba in these waterways. These waterways are an interaction between freshwater and seawater. And during these, in these 4 coastal lagoons, and we were able to examine the association between Acanthamoeba and water factors such as the oxygen in the water, and the bacterial species as well. So Acanthamoeba uses bacteria as a food source, so where we find bacteria, we generally find Acanthamoeba proliferating. So as I said, at Central Coast, 80 kilometres north of Sydney, we had 4 of these lagoons. We were able to look at the seasonal variation because we did it over 12 months with some slight delays due to COVID. We had 5 to 7 sites at each of the lagoons to get a representative of the area. This is a busy slide, but I just want you to look at the centre here, just in terms of our collection of the water samples. We filtered them within 2 hours, and it was this filter paper that we extracted, from which we extracted the DNA and then did downstream processing. So we had around 200 samples from the 4 lagoons, and we were surprised that there was such a high incidence of Acanthamoeba, remembering that these are, interacting with seawater, which has a high salt content. But this is the 38% is similar to what we found in dams in Sydney and also in domestic sinks. We've found an association with the summer months, increased Acanthamoeba with the summer months, and that is not surprising because Acanthamoeba, like many microbes, likes to proliferate in the heat. We didn't find an association with rainfall levels, and Acanthamoeba incidence has been found to increase after flooding. But we're having a very strange rainfall period in Australia at the moment, and so I think there's more to be looked at in terms of that. With the 4 lagoons, one Terrigal is more highly urbanised than the rest, and we found an increase in Acanthamoeba isolation at the Terrigal site. We're getting more incidences of these blue-green algae blooms in Australia, probably related to climate change, and they were positively correlated with the incidence of Acanthamoeba, as were a number of other bacteria. As well as Acanthamoeba being correlated with water temperature, we found this association with this gene, which is can be a proxy marker for people contamination of water. So stormwater runoff, where people are walking their dogs by beaches, etc., and you get that fecal contamination, which again is a food source for acanthamoeba. Most of the isolates that we found were the T4 genotype, which is the one that is most prominent in acanthamoeba keratitis. So it's important that we let contact lens wearers know that swimming is a risk factor for acanthamoeba and indeed for other infections, bacterial infections. But there are ways we can minimise the risk. There are a number of studies that have shown that goggles decrease the bacterial load on contact lenses, again speaking to the food source for acanthamoeba. And there are goggles available now that have lap counters, which you can get on Amazon, which are quite cool. There are other strategies around disinfection and disposability of lenses. And I'd just like to acknowledge on the left here, Binod Rayamarji, that this work is part of his PhD, and on the right, Professor Justin Seymour and his team who've collected these samples in the lagoons. Thank you.
Thanks, Nick. Time for a question if there's any burning questions. Nothing, you're off the hook, I think, Nick. Thank you. And we move straight on. So Christina's going to talk to us now about the potential of, sorry, where am I reading this? A potential new option for in-office disinfection of trial lenses. Thanks, Christina.
At the disclosures on the slide, I am a consultant for Ophtex Corporation, and my co-author, Katsuhide Yamasaki, is an employee of Ophtex, and we'll be discussing their products today. So I don't know how many of you recognize this scene, which is a bit of chaos in the lens disinfecting area, where you may have sticky notes, different kinds of cases. There's clear solution in it. You don't really know what that clear solution might be. So we are looking at ways that we can do a better job of creating effective disinfection for in-office reusable trial lenses. Currently, the recommendation by ISO and the CDC in the US is a full 3% peroxide soak for 3 hours. For gas-permeable lenses, they would then be rinsed in a multi-purpose solution, dried, and then stored dried, whereas soft lenses would then undergo a peroxide neutralization system in the standard case, required to sit for 6 hours, and then rinsed and then stored in a disinfectant case, which I think is an interesting and somewhat difficult thing to do. So it's a cumbersome and a situation where it's really difficult to tell what's happening at any given time. Povidone iodine is the solution that we're going to talk about today. Povidone iodine is very widely used in medicine, and it has been for over 150 years. It's used for hand disinfection, skin preparation, antiseptic irrigation, including ocular surfaces during surgery. We've also noted that there's a rise in the need for in-office disinfection because we're using more rigid lenses now and hybrid lenses with myopia control, including scleral lenses. And the PVPI systems, povidone iodine systems, are available in some countries throughout Asia, some of Western Europe, not in North America, and Australasia as well. But they are not approved or recommended necessarily for in-office disinfection. So wouldn't it be nice if you could actually tell by looking in a case very visibly whether it's in the process of being disinfected or whether disinfection is finished? So these are the two systems that are commercially available. The systems are slightly different for soft lenses and rigid lenses. So on the left in white, we have the soft lens system where you have a tablet that actually, it's HPMC coated on the outside, and then there's a layer of povidone iodine, and then the innermost layer is the neutralizer, as well as a protease, and it's mixed with a buffered saline. The rigid lens solution is a bit different. It actually has the povidone iodine already in solution, and the tablet that is added is actually the neutralizer plus a protease. This is the time course of disinfection for the soft lens system. So you start at 0, the tablet is in there, the surface of the tablet starts to dissolve, and the solution will immediately turn golden to orange. And then you have a concentration of about 50 ppm that exists over about 5 minutes, where you have full disinfection and you've got a darker color. Then as the neutralizer starts to be released, you see the color fading, and at the end of about 20 minutes, you see a perfectly clear solution, which means disinfection is actually accomplished. So in 20 minutes, you can have disinfection and a very visible indicator of when it's finished. The last 4 hours that are recommended are to let the protease continue working and to make sure any residual color in the soft contact lens has dissipated. The similar situation for the RGP lenses, except that it starts with full strength 50 ppm iodine in solution. The neutralizer then is dissolved in the solution, the color disappears, and again in about 15 minutes you have full disinfection and a clear lens that is showing you that disinfection has completed. And again, the remaining time of the 4 hours is to accomplish the protein removal. So if you look at are these efficacious, we can see here that the disinfection efficacy is excellent for the povidone iodine system. Within 2 minutes, it meets the ISO standard for a disinfection system, and within 5 minutes, all strains are below the detection threshold. You can see on the right hand what would happen if you put this in a peroxide system that has a neutralizing disc. So not nearly as effective in terms of time for the bacterial and fungal species. When we look at acanthamoeba, we're also seeing very effective disinfection of the trophs, and the cysts take longer. And even though disinfection has stopped at about 20 minutes, the iodine is gone about 20 minutes. Clearly, the trophs the cysts are damaged because they continue to die over time. And again, similar results for the peroxide system. So, could we, by using povidone iodine, make this confusing system a thing of the past? So, thank you very much.
Thanks, Chris. Staying on time again, but we can have time for a question or two if there are any from the audience. Nothing, I think. Thanks, Christina. Okay, our lucky last, Clara, is up now and is talking on the effect of crosslinkers on fluorohydrogel contact lens properties. Thanks, Clara.
Okay, hello. So my name is Clara Lim and I'm going to talk about the effect of crosslinkers on fluorohydrogel contact lenses properties. So as you know, the cornea needs oxygen to conduct its metabolic activity. As it's an avascular tissue, it relies on the oxygen flux coming from the atmosphere. That is why the oxygen permeability, also known as decay, is a key property of contact lenses. But there are also other properties that must be taken into account, such as the mechanical properties or the surface properties, so that we can have a comfortable and wearable lens. Currently on the market, most of the contact lenses are hydrogels. As a reminder, hydrogels are composed of long chains of polymers linked between them by bridges called crosslinkers, and they also possess a lot of water. So it is possible to modify their properties by playing on several parameters, such as the monomer's choice or the network type. But in our case, we decided to focus on the crosslinkers effect. To do so, we have synthesized a hydrogel, and we used the oligoethylene glycol dimethacrylate as the crosslinker.